with the first talk. It gives me great pleasure to invite my colleagues and friend, Professor Muhammad Sohail Sajid from the Department of Parasitology at the University of Agriculture in Faisalabad in Pakistan. Professor Muhammad, if you want to share your slide, please, you have 20 minutes. Thank you.
Thank you very much, Professor Sparagano, for my introduction. My name is Sohail, and I'm Associate Professor in the University of Agriculture, Faisalabad. And before starting, I would be thankful to the organizers for inviting me as a speaker in this wonderful webinar. I'm going to talk today about knowledge and gaps of vector-borne diseases and some scenarios in Pakistan. Most of the slides would be based upon my own research and a couple of other colleagues which are collaborating with me nationally. So the outline of my presentation would be vector-borne diseases, pathogens, and transmission, success tools for arthropods, then arthropod-borne diseases, the global scenario, and then prevalence of important vector-borne diseases in Pakistan, and then a brief outline of the projects which I have completed and are ongoing in my department, future threats and vector-borne diseases, and the roadmap to control them with future prospects in research in the country. This shows the vector-borne diseases, pathogens, and transmission. And this slide refers to a group of pathogens, means malaria, dengue, chikungunya, leishmaniasis, and Crimean Congo hemorrhagic fever. These are why I picked those, because these are prevalent and have been reported in Pakistan, and the vectors probably for malaria are Anopheles, and for dengue these are Aedes aegypti, and for chikungunya again Aedes aegypti and Aedes albopictus, and for leishmaniasis there's Leucemia and the flea-bottomless species of sandflies, and for CCHF this is Hyaloma ticks. So we have a diversified group of vectors which are capable to transmit a bundle of diseases to livestock as well as human beings. The success tools for arthropods, there are various molecules which are being targeted by these secretions which are present in the salivary glands of arthropods. These may be some vasodilators. These include platelets aggregation inhibitors, anticoagulation and fibrinolysis agents, fibrinolytic agents, I should say. There are complement inhibitors, immunosuppressants, or immunomodulators, and then wound healing and angiogenesis agents. So this group of the secretions, these are helping the arthropods for their successful meals. When we talk in case of mosquitoes, they are not feeding for a long time. We all know about it. But for ticks, it is quite interesting that they are happily feeding for a period of around 10 to 15 days. They stick on the body of the host, and they are all this success is due to these tools which are allowing the tick for prolonged attachment with their host. Then along with that, there are physiological tools. They do have exoskeleton, which is protecting them. There's a waxy layer. They have smaller size. They are capable to fly. No, not all, but a majority of those. They are having a very good reproductive potential. They are having a metamorphosis and adaptable to various kinds of climates. And diversification of appendages is there, which is making their ability to jump, to walk, to grasp prey. And in a nutshell, these physiological tools are equipping them for a successful a lifestyle in the environment, making it sure that they would be not only taking up their own meals but also transmitting the pathogens to the next host. So in the worldwide scenario is that there's a huge range of, this is not the end line. These only few of many which I have listed here. It includes dengue, Japanese encephalitis virus, West Nile, chikungunya, CCHF, Rift Valley, and sandfly fever. So this just to show the significance, that how significant these could be in the global scenario. And then coming down to Pakistan, we are also not capable to make it free from arthropods because of its geopolitical situation, and I should say that the transboundary nature of the diseases, because we are having the geography that we are on the north side, we are having China, on the east we have India, on the west side we have Afghanistan and Iran, and in the south we have Arabian Sea. So we do not presume that it should be free from such kind of vectors or vector-borne diseases because of transboundary nature of the pathogens, and we are having malaria, dengue, chikungunya, and a huge range of other diseases which has been recently reported by Ali, which are vector-borne in nature. The most significant bugs. The most significant bugs in Pakistan are these two, which are Aedes and Hyaloma. These are having characteristic features, as you can see on the right side of the slide. The top one is showing the tiger mosquito with specific white spots on the body, and the Hyaloma, which is called as the bont-legged tick, which is having the yellow rings on the legs. And these two are the most notorious bugs which are causing nuisance. The Aedes being the transmitter for the dengue, and chikungunya and Hyaloma for the transmitter of the CCHF. And both of these diseases had a recent outbreak in the country too. This is pretty busy slide showing the distribution of these vectors. On the left side, the map shows the distribution of mosquitoes in various parts of the country, and on the right side, the green map shows the distribution of various tick species prevalent in various parts of the country. So this slide shows that although we have a temperature variation from south to the northern parts, being some of the highest mountains in the world in the northern area and the coldest environment, but still the arthropods are very successful in making their lives success over there, whether it is a below zero temperature, or it is arid temperature, or it is a sandy temperature in the middle of the country, or it is kind of a wild desert kind of climate in the southwest part of Balochistan, we can say. So whatever the geography of the country is, they have well adapted to the kinds of climates. So a brief outline of the projects completed. I have started with my work back in 2002 with the brief, I should say, the classical epidemiology and associated risk factors of arthropods, which are infesting livestock and public health of Pakistan, which was primarily funded by Higher Education Commission. And then I got an opportunity to work on arthropod functional genomics, building community resources for animal health. It was funded from PAC US Science and Technology Cooperation Program. And then an offshoot of this project was exploring the ticks for rickettsial population within the country. And then I replicated the same stuff in mosquitoes because of their significance in the countries for finding out some mosquito species based on the genetic differences, as well as mosquito-borne diseases in livestock, as well as human side. A brief, some pictures related to the projects. The epidemiology of ticks, we worked on the 4 kinds of farming system in Pakistan, which are shown on the left side of the slide, which are rural subsistence, a smallholder production system, and then a rural market-oriented smallholder production system, and then below are the commercial dairies on a rural scale and the peri-urban scales on the lower panel. So all of these 4 kinds, we cannot say that these are devoid of arthropods in spite of their state-of-the-art facilities. We can say that the right lower panel, the peri-urban commercial dairy farming, can have the optimum preventive measures to get rid of the situations. Regarding the rickettsials, these graphs shows variable numbers, but in the nutshell, the cattle buffalo, means the large ruminants and the sheep goat, all of them, they were found infected with rickettsials when I identified based on outer surface membrane protein A and B based amplification through PCRs. Yeah, and this was the study which I published in Malaysia back in 2017. It was part of the project where we found that the ticks were infesting all the way from Mansehra to Gilgit. And for those who don't know where these are, these two cities are in the northern part of Pakistan, where there is extreme cold. And on the right side, I'm acknowledging these PhD fellows of mine. You can see the 9th highest mountain in the world, which is Nanga Parbat. And here, the unique thing was Jog, which is on the left lower side. It is from the bovine family, and it was also found infested with the ticks, and it was not found resistant. So it means that we do have a variety of reservoirs too, or maybe we can say the host which are available in the country for being attacked by the host, by the parasites. All right. So then arthropod functional genomics. Here the objectives were to detect the pathogens, and I should switch to the outline here. That what we did. We just reared the ticks on the rabbits using the animal institutional guidelines, and then we dissected for procurement of salivary glands, and for through first-strand cDNA synthesis, we went some proteins like VDI1B and we generated double-stranded RNA, and then that double-stranded RNA was induced into the ticks to have a knockdown effect of those, to see that what kind of phenotypic and genotypic alterations could be there if we block this VTI1B protein synthesis. And the reason why we picked this VTI1B was that this VTI1B was involved in the exocytosis process of salivary glands. So we just thought that if we just block this VTI1B, this orange colored structure sitting here, we may be successful in blocking this exocytosis process of the salivary glands, and side by side, we can also block the transmission of the pathogens to the host. So we found very good results out of it. But the problem is that there are some alternates too, which are taking role of VTI1B, and that is going on. So another gene we target is ferritin, which is a helping in the blood digestion process. So these results are underway, and we will be publishing them very soon. All right. So these are just results of transcriptional expression of VTI1B. And here, this shows that we knocked it down successfully. And then there was a major difference in percent attachment, mortality, and repletion of VTI silenced ticks as compared to non-silenced ones. And then this next project was on mosquitoes, where I replicated the same stuff. And in that project, and where we identified some gene genera and species which are prevailing in the country, but the results on pathogen transmission are still under process of publication. So they will be coming out very soon, that which kind of diseases are prevalent, which are carried by mosquitoes. Right. So another project is related to tick-borne ecologies in two different models of the country, of the province, I should say, in Punjab. And here I targeted some of the compounds like ivermectin and some biological compounds like entomopathogenic fungi. which include metarhizium and Anopheles against adult stages of ticks, and where I found that the egg hatching index, ovipositioning inhibition, and the death of adult ticks, and as well as larvae, that was based on these 4 parameters. This entomopathogenic fungi was working very well, at least at par with the available acaricidial agent like ivermectin. There are a couple of projects which are ongoing. It includes some geospatial distribution of arthropods funded by National Institute of Health, Islamabad. There is one project which I just showed the last slide. It is still ongoing on the biological control model of ticks and tick-borne diseases. Another project is Balochistan and Sindh provinces, which are funded primarily by HEC Islamabad. And then another project is also shortlisted, which is from the Endowment Fund Secretariat of University of Agriculture, Islamabad. And upshot about the outbreaks of vector-borne diseases recently occurring, not recently, it goes back to 2011 from the start of dengue outbreak, and then 2016 we have a reported case of chikungunya in Karachi, which is towards south of the country, and then in 2016 and 17 we had an epidemic of CCHF. In 2019 there is a dengue outbreak where 16,000 around cases were confirmed, causing more than 250 deaths in Lahore only, which is the capital city of Punjab province. Then in 2021, we have around 48,000 cases once again for the dengue. And now in 2022, we have 2 major threats, which is the lumpy skin disease outbreak in the livestock. And we still are not sure, but although the project that is recently shortlisted will cover this aspect too, that what kind of vectors could be involved in the transmission of lumpy to the livestock. And the second threat is right now we are going on, which is the flood. And that flash flood is attributable to 2 of the factors, which is the monsoon rains, plus there could be some climatic change and melting of the glaciers up in the north, which has caused which would be now leading to 2 major things, which is like mosquito propagation as well as other vectors leading to malaria and dengue outbreak, and already we are into these 2 outbreaks going on. So if we talk about the future threats of vector-borne diseases, it is very complex, and this paper, although it is very old, but it covers a bunch of factors which could be correlated with the vector-borne diseases. It includes climate change variability. It includes land use and deforestation as a factor, which can directly impact the vector, as well as it can go through indirect way through climate change and leading to vector propagation. It could be poverty, there could be water storage and irrigation systems, there could be sewerage systems, there could be international commerce and travel. So and this slide is not only referring to the country, but it is applicable to any part of the world, leading to the successful propagation of the vectors. Then how should we proceed? It's a good pyramid from Brax in 2011 that the roadmap to control vector-borne diseases it goes side by side. Like we should go for surveillance, and then we should also go for intervention on the left side as surveillance as in the long term in order to go for preparedness, as well as intervention to. So the future prospects for Pakistan would be like to establish a surveillance network for dangerous and emergent pathogens between government and academia entities in Pakistan, as well as this is very important to link globally at this point of time because of the threats of emerging and re-emerging diseases, especially of transboundary nature. Then we should also think of developing an online database based on proteogenome mapping, gene expression, as well as RNAi screen, or maybe including some other methods to go for their control. We should keep on identifying the potential vector species for arthropod-borne diseases using modern biotechnological tools, as one of the recent paper came from the KP province of Pakistan, where another species of heart tick has been isolated. And I just see yesterday that Emblyoma species is also reported from reptiles, which can further alarm the situation. Earlier, I have not found, I've just found one case, I think back into 1970s, that Emblyoma is reported here. So policy guidelines should be developed for disaster preparedness and management, because these disasters, these are linked with outbreaks and threats to, especially for vector-borne diseases. And then evaluation for livestock potential of Pakistan in preview of climate or disease resilient and resistant breeds should be done, so that the mitigation and adaptation strategies should go side by side for appropriate control measures. And finally, I would acknowledge all the funding agencies which have supported to conduct these kind of researches here in Pakistan. Thank you very much for everybody for patient hearing.
Thank you very much, Professor Sohail, for this exhaustive and informative and delightful presentation. You have shown so many data. I hope you will have some that you want to publish in this special issue. So we will be eager to read your paper and have you on this special issue. If you don't mind, we will ask the question at the end. So if you can stay, but if you still have power cuts, let us know. We will try to go around the problem. So if you can unshare your presentation, we will move to the presentation from Professor Leonardo, and then we will have the question for both speakers at the end. So
Makes sense.
Thanks, Professor Sohail. So can I invite Professor Leonardo again